Students hold different colored paper segments, each representing a part of the T plasmids, and begin to assemble them.
First, we have this red segment, our tDNA region. In its natural form, this part carries genes that cause tumors in plants, but for genetic engineering, this is where we'll insert our genes of interest. Next, the blue piece is the virulence or the Vir region. Think of it as the command center. It contains genes that produce proteins crucial for cutting and transferring the tDNA into the plant cell. Then there's the green segment, the opine catabolism region. This allows the bacterium to eat special nutrients called opines, which the transformed plant cells are tricked into producing. And finally, the yellow part is the region of replication. This essential component ensures the plasmid can make copies of itself, allowing the bacterium to pass on its daughter cells. These small paper strips we just added are the border sequences. They act like precise molecular guideposts, signaling to the Vir machinery exactly where to cut the DNA to ensure the only tDNA is transferred.
Based on our model, which specific region of this plasmid would naturally be transferred into the plant cell?
It's the tDNA region. That's the part that gets moved over.
Now, imagine the BIR region. Our blue piece became damaged. What critical function of the agrobacterium would be affected?
The transfer wouldn't happen. The Vir genes produce all the necessary proteins for processing and moving the tDNA, so if it's damaged, the system breaks down.
And why are those tiny border sequences we added so incredibly critical for this transfer process?
They are like the start and stop signs for the tDNA. They mark where the tDNA begins and ends, ensuring that only one precise segment is cut out and transferred. Nothing more, nothing less.
How might brilliant scientists modify this natural plasmid to achieve beneficial outcomes for humanity rather than just causing plant tumors?
They would remove the tumor-causing genes for the tDNA region and replace them with the genes that are useful to us, like genes for disease resistance, improved nutrition, or even drug tolerance in crops. Here are a small box symbolizes the Agrobacterium bacterium carrying its powerful T plasmid. The larger box represents a plant cell with this bead standing in for its precious chromosomes. They are currently separated just as a bacterium might be from a healthy plant. The student draws a plant signal card. This card indicates that the plant has been injured. It's releasing phenolic compounds, sugars, and experiencing pH changes at wound site, acting as a distress signal. The bacterium, ever the opportunist, detects these chemical signals from the wounded plant. This draws it closer, simulating the bacterium's attraction and attachment to the susceptible plant tissue. The red tDNA segment is carefully cut out from their plasmid bottle and attach one blue bead representing the VIR-D2 protein to it. The tDNA complex, guided by its protective VIR-D2 protein, now moves through the specialized secretion channel built by the VIR-B proteins, making its way from the bacterium directly into the plant cell. Once inside the plant cell's nucleus, the tDNA doesn't just float around. It successfully integrates into the plant's own genome, becoming a permanent part of the plant's chromosomes.
Based on what we just simulated, which specific components made it into the plant cell's genome?
Only the tDNA region was transferred and integrated. The result of the plasmid stayed in the bacterium.
And where exactly did this integration occur within our plant cell model?
It integrated directly into one of the plant chromosomes.
Now, if we hadn't modified this tDNA, what cellular changes would naturally result from this successful transfer and integration?
Naturally, it would cause plant growth tumors and produce opines. But because we can modify it, it could lead to the plant gaining valuable new traits like pest resistance and increased nutritional content.